I would now like to introduce Dr. Alessandro Sette back to the virtual stage to present the analysis tools. Thanks very much, Alex.
Okay, welcome back, everybody. What I'm going to do now is, again, share my screen. Okay, so what we're going to do is go for a broad overview of what the different tools are available in terms of analysis tools. So this is different from the predictive tools that you're going to hear more later. Analysis tools are defined as tools that can be used to further drill down in terms of epitope data that might be your own data or maybe resulting from query via IEDB database part. And Alba already giving you some very nice example of how different tools on the IEDB can be utilized for different analysis. So I'm just going to give you a more broad overview here. And this is the snapshot of the analysis tool page that you get if you go onto the analysis results and click on the analysis tools. And so there are different types of analysis tools. You see that there are some that have a green flask in the sense that they are tools that have been developed, but they're not completely fully validated. And so there is the plus to indicate that. So while we're still working to improve the quality of these tools, we felt these tools were quite useful and worked quite well for most applications. So we don't want it to delay their broad applicability for the community. So I'm going to just quickly go through the ImmunomeBrowser, and Alba already showed you several different applications of the ImmunomeBrowser, so I don't want to duplicate that. We can use the ImmunomeBrowsers to project user-defined epitope data or predicted epitopes. And as Alba showed, this can be used to look at, for example, spike and look at three-dimensional structures, antibody T-cell reactivity. So it's really broadly applicable analysis tool. An analysis tool which is very popular and is used frequently is the population coverage tool. So this relates to the fact that different HLA alleles may bind or be restricted to the different epitopes may bind or be restricted by different HLA alleles, and these HLA alleles have different frequency in the general population or in different ethnic groups. And so it becomes interesting to try to project what kind of coverage you get. What fraction of a given population is covered by a particular set of epitopes with defined restriction or binding capacity, how many epitopes bind, and so forth. So the tool does that. So you can select different population from the HLA frequency.net database, and the tool will tell you, for example, here in this example, you see for a particular set of fictional epitope in this example, we say how many people in the Algeria population would bind this set of epitopes. And so you see here, this red line gives you the 90% coverage, so tells you how many people would be covered. This line gives you the number of hits, and if you want, here it's 50%. So in this particular population, you would say that the average person recognizes 9 different epitopes from this fictional set of epitopes that we define. Now, there is one issue with the HLA allele frequency net is very granular, has a lot of different population. One issue, though, is that for some population, the data is incomplete, so it looks like there is no coverage, while in reality, there is no data, which is a quite different thing. And also, a lot of times, the HLA allele frequency.net resource gives you the HLA frequency of the ethnicities that are ancestrally related to that population. So, for example, you might get that a particular geographical location has HLA frequencies of the indigenous population. So, for example, if you go into Australia, for example, you will have the Aboriginal frequencies, but if you are looking at a clinical study in Sydney, you will have today in Sydney, there is a lot of still Aboriginal origin, but also a lot of immigration from different localizations and so forth. So to address this issue, we also including now a different source of HLA frequency in different ethnic groups based on transplant marrow donor program databases. So that is that HLA frequency is more reflective of the general population living in the US donor registry that also declare their ethnic origin. So in a given ethnic origin, so for example, Vietnamese ethnic origin, then you will know what is the frequency of all different HLA alleles. And we will maintain both sources because for different application, you may be interested in different sources. And so this is one way the data can look like if you plot it in a heat map where you have all different ethnicities or geographical locations. This is a number of different set of epitopes derived from different ORs for class 1 and class 2 from SARS-CoV-2. But just gives you an example of how you really can use this tool to get population frequencies for different ethnicity or worldwide for a number of different epitope sets of interest. Another set that is widely used is the epitope conservancy analysis. And so here, what you have, again, is your set of epitopes that may be derived from your own research or maybe derived from a search of the IEDB database. And you want to ask how homologous this epitope or epitopes are to a set of protein sequences that, again, you're providing from having queried different databases, or maybe it's your own data, and you specify a certain level of homology. So you say how, here you see in this particular case, you can say 100% or 70% or 80%. So you can see and filter based on that level of homology how many hits you have for that level of conservation. So to give you an example, we are interested in arenaviruses, which are a family of viruses of potential pandemic concern. And there are different types of arenaviruses. The one that is perhaps a prototype virus is the Lassa virus that causes very serious hemorrhagic fever and deaths and is endemic in West Africa, and it's a constant problem there. There are other arenaviruses that are frequent in the old world, so Europe and Africa, such as L3MB, Mupeya, and so forth. And then there are a number of other viruses from the arenavirus family that instead called the New World species in essentially South America, and these are some of his names: Tacarigbe, Guanarito, Junin, Sabia, and so forth. And so the question becomes, if I were to develop a vaccine based on Lassa, would I get coverage of all different Lassa strains? Would I maybe get coverage of all old world arenaviruses? Would I maybe able to expand my coverage to new world arenaviruses? And this is data that has been generated for epitopes that we have defined using the and we analyze the conservation using the epitope conservancy tool, and you can see that these various epitopes we have defined are very conserved in Lassa lineage 4, which is a prototype Lassa virus, also in other Lassa virus of other lineages, and even in other old world arenaviruses apart from Lassa. So this suggests that you can indeed perhaps develop a T cell component that is pan-reactive with all old world arenaviruses. But when you look at the conservation of these sequences in new world arenaviruses, the conservation really drops down, and there's only a couple of peptides that are significantly conserved. So this is an example of a type of analysis you can do using the conservancy tool. Cluster analysis tool is another popular tool, and this the rationale for this tool comes from the fact that you might have data. For example, if you query the IEDB, you have data from a particular protein from a variety of different studies, and people may have used not exactly the same peptide, overlapping peptides or peptides with different sizes, but overlapping. So you want to be able to cluster the data to distinguish instances in which maybe the epitope is slightly different, but it's substantially the same region of the protein. And again, you can specify a particular identity threshold, and different approaches are identified, basically discriminating whether the tool, the cluster that is defined is non-redundant, meaning that each peptide can be only found in a given cluster, or that depending on the homology, the same peptide can be found in different clusters that are partially overlapping. And these will be also described in more detail tomorrow. Finally, on the lab grade tools, the RATE is another popular tool. And this is a tool that is useful to infer potential restrictions. And this is something that in the field of HLA is done all the time. You're probably very familiar with the data that suggests that particular HLA-DR4 alleles are associated with rheumatoid arthritis, or particular DQ alleles are associated with diabetes, and so forth. So essentially here, what the question we're asking is the reactivity of one particular peptide or set of peptide particularly associated with the presence or absence of particular HLA types. And this can give you really a hint in parallel with the binding capacity with which particular HLA molecules may be actually the restriction elements for that particular peptide. And so this is an example of this output. And so for this particular peptide number 1 with this particular sequence, you can see that there is an association with HLA-DR beta 30202. And why is that? Because if you look at the distribution here in these 50 donors, that have been tested for activity to this peptide. Again, this is a fictional data, or actually maybe real data from one of our papers, but it doesn't matter. This is to illustrate the logic here. So you have these 4 different categories. A means HLA, and R means response. So in this particular case, you have that 6 people expressed this particular DR beta 30202 HLA molecule and were also responders. And in no case you had a responder that was negative for this DR beta 3 molecule. Of course, restriction is not absolute. You would have also that some people express the HLA in question, but did not respond. And finally, 26 alleles, 26 individuals did not respond and did not have that particular HLA molecule. And so you can calculate based on these numbers, essentially a Fisher exact test, and you get values of p-values that are associated with the projected restriction. And again, this is a projection based on HLA frequency, and this can be then coupled with assessment of a predicted or measured HLA binding capacity, and eventually can make tetramers to verify the validity of a projection. There are also tools that allow to look at immunogenicity predictions, and one popular tool is the Deimmunization tool. This tool is useful in the case where you, for example, want to reduce the immunogenicity of a protein. For example, a protein drug or a non-fully human monoclonal antibody, but is a drug that is used, injected into patients. You don't want to have an immune reaction against the drug because that could create safety problems and minimally reduce the efficacy of the drug. So you can use this tool to select predicted epitopes, and then the tool will calculate the predicted probability that an analog where a single substitution has been introduced would have an impact on the binding capacity for the general population. And of course, you want to have mutations that decrease the general binding capacity. And again, this can be used then in conjunction with classical medicinal chemistry analysis, because you want to introduce mutations that would decrease the immunogenicity, but without interfering with the drug biological activity and pharmacodynamics and so forth. Another tool that is of general applicability is this PepSySco that, given a set of peptide sequences, will give you an estimate of how easy is this peptide going to be in terms of synthesis, alerting you to maybe you want to use particular care or have a separate synthesis for this peptide. And it's known that in general, the peptides that have stretches of repeat aliphatic residues are particularly difficult to synthesize. So this, just looking at this guy, this is going to be very difficult to synthesize, and you get the score that tells you exactly how probable is that this particular peptide will encounter synthetic problems. And finally, PepX, it's a peptide expression annotation, and this goes back to some of the discussion we have had already in what other features beyond predicted peptide binding capacity may influence potential immunogenicity, and certainly the level of expression of a peptide is one of them. So it stands to reason that a particular peptide may have will have certain predicted binding affinity. But if a peptide is expressed in a protein that itself is made in very small amounts, this will have a lesser chance to be bound in significant amounts and recognized by the immune system in comparison to a peptide that would have a same sequence but be found in a protein that is expressed at very high level, perhaps a structural protein. And so same thing here, you enter your sequences, and then the tool will look at expression level of most peptide from selected public databases and return your results. This is a recap of the various different tools that we briefly covered. The ImmunomeBrowser to visualize and plot reactivity along sequence protein, the population coverage, the conservancy, the clustering rate, the deimmunization, and the PepSySco and PepX, more recent tools to predict synthesis capacity and expression level. And with that, I will end this presentation and turn to the questions.
Awesome. Thank you so much, Alex. I'll give you a couple of minutes to have a look at the Q&A, and then I will invite Alba back, who is valiantly fighting through the questions because she's not doing too well, but thank you so much. So Alba, take it away.
Thank you. So the first question is, Alba, thanks for presenting your recent work on SARS-CoV-2. I'm curious if in your analysis you found any evidence showing that CD8 T cells can kill a subpopulation of B cells that present SARS-CoV-2 epitopes. That's a very interesting question. We have not looked into that specifically. With the bioinformatic analysis, it's challenging to understand what type of cells is going to present the specific epitopes, but that will require specific experimental validation that we haven't looked into that. Happy to discuss this further, maybe offline. Alex, you want to answer the next one for yourself?
There are two questions from one from Mauricio and one from an anonymous attendees that ask whether PepX consider expression only under steady state conditions, what inflammation of a protein is regulated, and that's a very good question. In general, PepX relies on public databases, so that may be a mix and match of what is available. Certainly, the expression level in different conditions, I agree, it does matter. And in fact, for example, there are studies to suggest that CD8 cells may recognize early viral products that are expressed early in the infection, while CD4 tend to recognize more structural proteins that are expressed later in the infection, and so forth. So it's a very good question. And the other question is an anonymous attendee that asked whether PepX return just to full viral expression, or it would do some differential gene analysis in the background. In general, PepX, I believe it's just focused on mRNA expression. I think that there is not a lot of data really reliably looking at protein expression. Alba, do you want to take another one?
Yes. So Kieran, thank you, Alba, for the insightful talk and case study. I was wondering if any 3D molecule studies such as protein peptide docking were conducted. Thank you so much for your kind words. No, we have not been doing 3D molecular studies. You're also asking what type of parameter can be done. So I totally recommend you to follow next day workshop where they're gonna discuss a little bit more the tools with the TCR matching and so forth, but we haven't done any of those in our studies. Alex, you wanna go to the next one?
Yes, Mauricia was asking some questions about the p-value for the rate tool. And in general, yes, so there is a p-value that is essentially the p-value of a Fisher exact test. But after saying that, the issue is a bit more complicated than that, in the sense that in the publication, we emphasize that that p-value is not Bonferroni correct. And for those of you that do HLA frequency association, that's an important issue, because basically, if you have in your population expression of hundreds of different HLA types, just by chance, something may turn out to be statistically significant in the Fisher test. But if you do a Bonferroni correction, that might not be significant anymore. We chose to put a non-Bonferroni corrected value because there are many different ways to do Bonferroni corrections. You actually can select the more frequent HLA types to reduce the threshold of Bonferroni required, because in any case, if you have an allele that is expressed only in 2 people in your population, you can't really make a meaningful association. So there's a p-value, but that should be done keeping in mind that those values are still likely to require experimental validation.
Alex, I think the last question are all for you.
Okay. There is a question about okay, hold on a second. Okay, so there is a question from an anonymous attendee. Sometimes I see tools use name like MHC binding prediction or MHC presentation prediction. Are they the same from a biological perspective? They are not. And the binding prediction is, technically speaking, expresses the likelihood that that particular peptide will bind to that particular HLA. And that is also a quantitative measure, and you can actually predict, and we will get to that in the analysis tool in much more detail, but you can predict a binding affinity. The presentation refers more to studies where the tool is trained on elution data. The elution data, it's great because there's a lot of it. And also, in a way, you would imagine that in an elution prediction, the additional variables, such as the capacity of that peptide to be generated by processing, is also accounted for. A drawback of elution data is all or none. So either the peptide is present or it's not, or actually there is no none. It's only telling you that this peptide was found. So usually there is an issue of generating good set of negative data to train algorithms and things like that. And in reality, the observation has been that the accuracy in terms of predicting eventually T-cell recognition is highest when you combine both elution and binding predictions. there is a question to show some example how to determine immunogen or epitope about a cancer biomarker, and I would say that that is relevant to for the third day when we will actually look at the CEDAR database that is focused on cancer application.
There is a question from Massa with a specific
Yeah, I wanted
sequence
to take that.
Yeah, that one I know. With a specific sequence of peptides, because I've been doing that a lot lately. With a specific sequence of peptides, part of the spike, would it be possible to assess the degree of conservation across all known SARS-CoV-2 variants in IEDB? So the way you can do this type of analysis is using the conservation tools available in the IEDB. You can add the specific sequence of your peptides, and you can use as the sequence readout as many SARS-CoV-2 variants as you're interested in, and the tools is going to be able to provide you an average conservation, or even calculating the conservation for each one of the isolates independently.
So then there is another so there is a question, why is negative data much less than positive in the IEDB? Well, because people don't report negative data. And so we can only curate what people report. And it is indeed indeed a limitation, and that's why also NIAID has been very prominent in for the direct submissions, asking people to also submit negative data. There is a question about the tools. What is the likelihood of homology between the human proteome and the predicted epitope? How reliable is the assessment in terms of ensuring safety? Well, I mean, you can certainly blast an epitope against the human proteome and see if there are hits that come out. In general, I think this is informative, but not probably very reliable for a couple of reasons. One is that autoimmune cross-reactivity and mimicry has been reported, but there is really not a good strategy to really select what is a significant level of homology. And also we've seen that you can get cross-reactivity at the T cell level with very variable degrees of homology. Sometimes you can have even a single substitution completely obliterates T cell reactivity, and in other cases you can have cross-reactivity with what superficially looks very minor homology. Then there is Salman Ali asks, in the epitope conservancy, the first step and the second step, and the answer is yes. Yes, you have it correctly, I think. What I see threshold value for MHC 1 and 2 binding, I think that's to be answered tomorrow in the context of the that. So it is correct to assume the entire epitope content of a CEDAR database is still found in the IEDB database. Yes, in that respect. So there is a overlap between cancer epitope and non-cancer epitopes. And there are two different overlaps. One is well known that certain viruses are actually linked to cancer. So HPV is linked to cervical carcinoma, hepatitis B and C are linked to hepatocellular carcinoma, and so forth. So in CEDAR, you will see that there is an option to also select for those viral epitopes from viruses that are known to be associated with cancer. Conversely, it is there are self-sequences but are not mutated, and they could be recognized in absence of cancer, absolutely. And there you go in a little bit or more of the overlap, in a way, self-reactivity. So reactivity against a non-mutated melanoma antigen, for example, or against CEA or other non-mutated antigens. Well, that could also be considered autoimmunity in a way, right? So autoimmunity is immune reactivity against a self component. And so that there is another area of overlap. I have a question about the source of epitope sequence. If an assay is done with a longer peptide antigen, how is the epitope residues being defined? Again, this is, the IEDB largely goes by what is reported by the authors. And certainly there are different situations that originate. So as I was introducing the database this morning, I say, so we set an arbitrary threshold of size of 50 amino acids. So someone may report 50 amino acid stretch because I have shown that those 50 amino acids are associated with T cell reactivity. That doesn't mean that all those 50 amino acids are the epitope. In fact, there may be subsequent studies that will narrow it down to a particular 15 amino acid fragment of that 15. So that is a situation that we refer to as an epitope containing region. Conversely, there are studies where maybe someone has defined and mapped very precisely the epitope, and so that is the exact epitope. And this is, for example, is a situation where in the antibody situation, you have a crystal structure, when you know exactly what the epitope is that is recognized by the antibody. Then there is a third situation where there is incomplete knowledge. So say, for example, that someone has performed a series of studies with viral mutants that escape recognition from an antibody. And so you might have mapped 3 particular residues that have been shown that when that residue mutates, you lose recognition from the antibody. That doesn't mean that only those 3 residues, that's a complete epitope. So those are residues inside the epitope, but the actual epitope may be, and usually is, longer. So, and last question is, are physicochemical properties such as hydrophobicity, hydrophilicity, charge, and polarity important? There was a lot of literature trying to predict, and this is where it goes back to the early 80s, where people were using hydrophobicity, hydrophilicity, amphipaticity to predict T-cell epitopes, and that really never worked because it doesn't really take into account the fact that different HLA bind different motifs. So that really, for T-cell, did not further develop very much. For antibodies, still there is some success of algorithms that, again, are very old to predict antibody reactivity based, for example, hydrophilicity and charge. And that is basically because antibodies tend to bind to things that are on the outside of proteins. And so usually or frequently, proteins have a more hydrophobic core and a more hydrophilic outer. And so by predicting hydrophilic stretch, you have some degree of prediction of antibody reactivity. But the more recent antibody predictive tools have expanded beyond that. And I think you will hear more about that tomorrow.